Today I will just introduce a publication that we recently published in Cell Host, then in the strain level genetic heterogeneity and the colonization dynamics drive microbiome therapeutic efficiency. Okay, there's a very brief introduction regarding the cancer and the microbes. About in 2020 or 21, there's a lot of publications saying that we find microbes in different sort of cancers with different composition of species. And at the meantime, we also see that a lot of publications regarding how the gut microbiomes can indirectly influence the development of the cancer. So that means that instead of pure human-specific process, that microbes also contributes to the development and possibly the treatment of cancers. So about the same time, there's a lot of publications regarding how we can actually use those microbiota transplantation to treat or to help the treatment of cancer. They start from this melanoma and recently got lots of new publications in different kinds of cancers. But I think there's several key questions that hasn't been resolved. One is that which bacteria can be transplanted. So most of the analysis are focused on species level. That means that you cannot actually see that whether the specific strain of the bacteria has been transplanted from the donor to the recipient. And also we do not know that which bacteria can influence the treatment efficiency. So not all bacteria are the same. Some can be beneficial, some can be deleterious. So we need to know that which specifically actually change the treatment itself. And can we actually identify the key bacteria so that we can design a new composition of bacteria rather than using those nature fecal microbiota for the transplantation. My research starts with a clinical trial. This is a single-arm clinical trial targeting the lung cancer cohort. So we use the FMT and plus immunotherapy. We do 5 times of FMT in total, and add in this PD-1 treatment and also chemotherapies. Actually, the enrollment of the cohort is not that great. We target for 30 patients in total, but at the end we only got 14. And 3 of them actually dropped before finishing the FMT process. So they are not included. So at the end we only got about 11 patients. Luckily, most of the patients, they actually got a quite good response. We got around 60% of the stable disease or complete or partial response. So there's a very high level of responses comparing to the control. The one problem is that although we can get controls from its previously published clinical trial, due to the single-arm design, we actually cannot compare our patients with the patients that using in the same environment directly. So we decided to go to the city center and find the government. We actually found that they actually got lots of established records. So we managed to build up synthetic reference control. So with about 38 patients, they are taking a similar treatment but without FMT. We see that we actually doubled those progress-free survival time period from only 6 months to 12 months. And we also got one particular patient. He actually got a very significant reduction of its lung cancer and up to over 3 years now. So it's a very good response. However, we also noticed that due to the limited number of patients we enrolled, we haven't got enough samples to be able to set up any meaningful results of its own. So what we decided to do is that we can find lots of public cohorts, although with different diseases. We hope to be able to find whether there's a similarity between different diseases. Then we built up a healthy core microbiome. So in this study, we got 3 FMT cohorts and 3 probiotic cohorts and 1 longitudinal monitoring cohort of healthy individuals. The first thing we find, which is not very surprised, is that there's no single bacteria that actually being beneficial or being disease associated across all the cohorts. The reviewer says there's no surprise. You have so many different diseases. It's quite okay. So to be able to identify what is actually being changing between healthy and the disease beneath this species level, we actually designed a whole new pipeline. Actually, in the previous talk, somebody discussed about the co-genome MST. I'm the main developer of CGMST, so I decided to actually use this bacterial genome method and move it into the microbiome study. So in this our study, we actually set up a whole set of universal cogenes, 480 universal cogenes that covered all tree of life. We got about 173 cogenes covering bacteria and 200 covering archaea and 380 covering the fungi and the parasites. So this set up a whole set of cogenomes which covers around 100 kb regions in the genome, covering about 2% to 5% of genomic region. That means it actually gives us enough resolution to differentiate strains that's with over 1 in 1,000 sequence differences. So based on that, we built up a large set of co-genome reference data set. Use all these over 1 million genomes in RIFSEQ. We screen them, filtering those only leaves the sequences with over 2% sequence differences. So at the end, we managed to reduce the total size of genomes by over 10-fold, and due to the use of CGMST, it's further reduced the genomes, the sequence database size about 50-fold. So at the end, the final database size is only 9 gigabytes. That means the whole database can be transported easily. Okay, we designed our pipeline based on these cogenes, and one thing is that it can be used for taxonomic profiling. So we compare its performance with Kraken and with MetaFrame, and found that it basically a little bit better than both of them, but not that significant. The most important part is that due to the use of cogenes, we can reconstruct the cogene sequences of each strain. We compared the performance of our pipeline with the other assembly-based method of strain frame, and found that it can reconstruct lots of many new strains from the metagenome. And we evaluated the performance of our method in those probiotic cohort, and found that the probiotics you take can actually survive in your gut for over half a year. And we also evaluate the tree in our patients, and found that for some probiotics like AKK strains, it can actually see a direct transmission from the donor to the recipient. That means our method can accurately check the strain transmission due to FMT, but for some other species, we cannot check the transmission. So that means that we have some variations in terms of their performances. So the main question is why this happens. We found that it's actually been intrinsic characteristics that whether a bacteria can colonize in gut or not. We found that about 47 species that can persistently colonize in the gut, and they can survive for over half a year and up to 2 years in our cohort. But we also have some bacteria that can only localize, persist in the gut for very short time. So it will lost within 1 month. We analyzed the different characteristics and found that the gut-adapted species, they tend to have a very component immune evasion capability, and also they are good at anabiotic respiration, whereas those rapid turnover, they got a very high mobility and they got a highly variable nutrient utilization capability. That means they can survive in different environments, not necessarily only survive in gut. Okay, another question that's been more interesting is that we would like to see whether we can find some strain-level disease association. There has been a lot of publications in that field. For example, in the Cell Host paper, they found some bacterial clades. They have a association with IBD, whereas it's associated with the healthy individuals. Similarly, in this Nature Medicine paper, they found the type 2 diabetes also associated with different clades in 2 different species. So in our study, we managed to screen all these different species and identified over 100 species that's highly associated with healthy or diseased lineages. We can show one example. Oh, not there. Okay. In this figure for Faecalibacteria duncanins, we see that in the top part, we have these disease-associated strains, whereas in the latter part, we see a lot of healthy associated strains. That means that within one species, we can see some clades being healthy associated, one being disease associated. Using those 108 species, we designed a machine learning model and a score called HSIN. That means the scoring of healthy strains. We managed to effectively predict population status, means whether it's disease or not, with AUC of 0.8. Okay, so that's a summary of my talk. We developed UC-GenomST. It's actually been an extension of C-GenomST and being used in metagenomics. And we found that FMT success relies on strain-level engraftment or clearance. We found bacterial colonization is an intrinsic trait with distinct ecological strategies, and strain-level specificities drives health disease associations, and healthy strain proportion is a robust predictive biomarker. And thank you. That's my talk.